So Dr. Turner has a very interesting background. Was an undergraduate at WashU, and then was an MD PhD student at Case. Came to Boston, like many of us, for residency and research. It was at the Brigham, or pathology residency. He's a full professor of pathology and a full professor of medicine at Harvard Medical School. Then spent actually the bulk of his career at the University of Chicago. I'm a proud UC parent. My son was an undergrad at the University of Chicago. And actually in 2017 came to back to Boston, which happens to many of us. I was away for a 20-year period and then sort of cycled
It's like a vacuum, sucks you back.
There is. He was associate chair at the University of Chicago in the Department of Pathology, and actually the editor, associate editor, editor-in-chief of really some of the leading journals in the field for many years, Laboratory Investigation, and actually the American Journal of Pathology, which is, I think, the leading journal in the field of pathology.
That's one of them, yeah.
One of the leading,
yeah.
And also editor-in-chief of what, molecular and cellular...
Other way around, cellular molecular.
I'm sorry.
It's all right. It's we just say CMGH, it's a mouthful.
Okay.
And the MGH guys really like that.
So Dr. Turner is a leader in the field of inflammatory bowel disease and really understanding gut barrier integrity. And I think, like each and every year, the sort of focal point of the gut being at the axis of everything in life, whether it's the brain or other components of our physiological system, especially with the microbiome playing an increasingly important role, is front and center. And so Dr. Turner has really led the field in understanding the role of intestinal and gut barrier integrity in health and disease. So again, it's great
Thank you.
To have you over here. Yeah.
Thanks very much for that kind introduction. So,
and actually, so we have a broad group of research faculty and students, surgical residents, and part of why we have this seminar is to expose residents who are both in the lab, but those who are also thinking about potentially research fellowships of really potential great mentors, in the Harvard community. So again, thank you.
I hope I'll convince you there's exciting stuff going in the lab. What Dr. Shagoff said is right. You can actually live okay without much of a brain. We see people in the streets all the time like that, but without intestines, you're stuck. So really, this is the most important organ. So my conflict of interest, as you'll see by the time I get to the end, we've developed something that we think is a proof of concept molecule for a therapeutic, and so we've started a company to try and pursue development of small molecules that could actually be that therapeutic. The molecule I'll tell you about cannot. And so I'm a co-founder of that, and but I'm not going to talk about it as therapeutic use because I would never ever use it in a patient. It's not there. So tissue barriers are really required for the survival of multicellular organisms. These are present throughout your body, lots of places. So we all know about the blood-brain barrier. The intestines, you've got this single layer of epithelial cells really separating you from the dense microbiome on all the things that are in the lumen. That's really key that be present. There's other barrier sites that we think of. Tight junction protein mutation animals often show up with male sterility because the testes blood barrier is damaged. The inner ear is another place where barriers are really critically important. But it's really a primordial function that things as ancient as sea sponges make barriers. If you want to be a successful multicellular organism, you need to be able to partition, and the Blues Brothers line, there's two kinds of people in this world, us and them. You need to be able to tell the difference. So the intestinal epithelium provides that barrier between the external environment, which the gut is really a tube of the outside world going through your body if you think about it anatomically. And really, there are cells, and that single layer of cells is critical. But if you think about it, when the cells touch each other, there's a space. And that's called the shunt pathway. And things could just go through there. So you could just go around the cells, and there wouldn't be much of a barrier. So really, it's the tight junction that's the key to that. This in a transmission, EM, is the apical junctional complex. So you can see microvilli up here. These are intestinal epithelial cells. This is the tight junction. It's fairly nondescript. The adherens junction is maybe a little bit more interesting. and the desmosome, which often people point to and say that's a beautiful tight junction, just because it's so much prettier than the tight junction. The tight junction makes up for it in freeze fracture, where it's this beautiful stranded structure, and the networks vary in density and how many layers of strands there are. We don't fully understand them, but there's a whole nother arm of my lab that I won't talk about the work at all, where we're studying some of the proteins that make ion channels in these strands, and really how ions and water flow across the tight junction. I really won't talk about that at all today, but I'm happy to answer questions. On keyboard. Okay, so before I start, the take-home message is, I hope you'll come away with, is to understand that tissue barriers are regulated in response to physiological and pathophysiological stimuli, that barrier regulation can occur via tight junction dependent and tight junction independent mechanisms. I think, as I'll show you, this is sort of obvious, but it's so obvious that a lot of people who are writing papers don't think about it, and they ascribe every barrier defect to tight junctions, which just isn't the case. And then hopefully I'll convince you that detailed mechanistic understanding will lead us to the ability to target molecular interventions, and that can have incredible promise as non-immunosuppressive, fairly benign therapies. So I love this slide. I took this off the internet, obviously. I don't know Karen, but I really like the way she thinks, because she thinks that intestinal permeability defects are responsible for 90% of all chronic symptoms and conditions. So this will be the most important seminar you hear this decade. I tried to use this line in NIH grants because I thought that would get me funded, but it didn't really seem to work. So this is excessive, and actually if you do a Google search for leaky gut, I hope you're horrified. But I'll show you data related to that. It's not a disease. It's not a syndrome. It's not an anything. But I'll show you some brand new data that is under review right now, not yet published, where I think there might actually be some truth to this thing that I thought was bogus for so many years, but not the way they say. Okay, so these are the sorts of diseases, and I don't know if you can read this, but I can go through some of them. These are the sorts of diseases that people have implied have barrier defects, and this is not an exhaustive list. Pretty much everything under the sun, people have said, oh yeah, chronic malaise, barrier syndrome, chronic fatigue syndrome, barrier syndrome, Gulf War syndrome, barrier function. So everything you can think of. But there's really pretty good data in terms of correlates with disease in inflammatory bowel disease, graft versus host disease. In graft versus host disease, it's just a correlation. We don't really know if it causes it, and actually, since the gut is a target of graft versus host disease, it may just be telling us that when disease is worse, there's more gut damage and more barrier loss. So we don't really know. In inflammatory bowel disease, if you take patients in remission, I'll show you the data in a moment, it can be increased permeability can be a marker of disease relapse. So there's some evidence for function. Everywhere else, type 1 diabetes, there's often an increase in before diabetes develops. HIV/AIDS, there's an increase in permeability. People think that might be responsible for the increased cardiovascular deaths in HIV patients who are on antiretroviral therapy that's successful. They still sometimes have permeability defects. We go down celiac disease, irritable bowel syndrome, food allergy. Under some conditions, in some patients, there's barrier defects. And then you can go on autism, eczema, psoriasis, fibromyalgia, depression, rheumatoid arthritis. You can go on, and there's less and less data in various things, but they've all been pointed to that. And I think one of the key problems is we don't really know how this intestinal barrier works. We know it's made up of cells. We know the junctions are important, but that's about it. So I'll talk about IBD as probably my central example, but I'll talk about graft versus host disease a little bit too. So this is, these studies were done in Crohn's disease patients because Crohn's disease patients, we can use the permeability probes. The ones that are used here get metabolized by colonic bacteria, so you can't really use them to measure colonic permeability. But this is the lactulose mannitol assay that maybe some of you have heard of. These are two non-digestible sugars, and we don't have transporters for them. And so if they get absorbed into the bloodstream, it has to be paracellular, it can't be through the cell. And then they get filtered in the glomerulus and end up in the urine, and we don't have any way to reabsorb them because we don't have transporters for them. So they're an indirect marker of intestinal permeability. You have to make some assumptions, things like renal function is intact. But they work pretty well. And this is, the first couple of columns of this are replicates essentially of a study published over 30 years ago. The last column was added, and the whole graph is really from a study from almost 15 years ago. So you can see the field hasn't moved forward really at all beyond this. When I was trying to decide on a focus of research, this was one of the types of studies that really convinced me that this was something I wanted to work on. So we had this observation that if you take healthy controls and Crohn's disease patients, some of these patients have increased intestinal permeability or reduced barrier function. Now, in some cases, that might be due to frank epithelial damage, but sometimes not. But the striking thing is if you look at their first-degree relatives who are healthy, these are healthy people, a subset, about 10% of them have increased permeability. So that tells us a couple of things. And the added column more recently from the Buhner study tells us that it's not environment. It's not the microbiome, for example. These are non-related cohabitating people. They don't have this. And in fact, the Buhner study linked it to specific NOD2 polymorphisms. So this tells us that normal people, healthy people, without any apparent disease of any sort, can have barrier defects. All right, so right away that really throws a lot of dirt on the leaky gut syndrome idea. Okay, here's the other part of data that I think is really interesting. This is one of at least 3 studies showing this. At the beginning, these are Crohn's disease patients. They were all in remission. They measured permeability and followed them. Those at time 0 with increased permeability, a ratio greater than 0.03, had a much higher rate of relapse. You could say, oh, well, they were about to relapse anyway. You just caught them as the damage was happening before it was clinically evident. But that's not true. They relapsed over the course of a year. So what does that really mean? The final thing that I'll tell you, there's no published data yet, but there's unpublished data. We have wondered for 30 plus years whether those Crohn's disease patients, whether those relatives with increased permeability are at increased risk of developing disease. Would those be appropriate people to target with preventative therapies? Nobody ever did the study. These are hard clinical studies to do. You find a family, you test them all, you find some with permeability defects, some without. You say, I want to follow all of you for forever to see if you develop disease. Finally, that study's happening across Canada mostly, and the data the rates of conversion to disease from non-disease are very low, but it looks like they're quite different in people with increased permeability. So it's looking like that actually might be a marker of impending disease. It still doesn't prove that it's causing disease, but it's certainly a marker. I decided that really the way I would try and understand this was first to understand something about how barriers are regulated. So at the time I got into the field, we didn't know anything really about tight junction regulation. We knew if we depolymerized actin, we could mess them up. And that was about it. And there was this one guy, Jim Madera, who had done studies like this where he showed that, well, let me back up. I'll talk about John Medding's studies first. So this is a study from John Medding's, but it's based on Jim Madera's studies. Jim Madera was here, John Medding's is in Canada. The green line is what you'd expect for sodium nutrient cotransport, transcellular, right? It's saturable. We all understand that. Unfortunately, that's not what you see when you do an experiment in rodents. You get more and more D-glucose, and you get more and more absorbed. All right, so the x-axis is how much is in the lumen, the y-axis is how much is absorbed. But you see there's an inflection where the slope of the curve changes. And that's kind of a hint. If you give L-glucose, which is not recognized by the transporters and is not transported across cells, but only paracellularly where there's not that molecular recognition, you see that L-glucose is a straight line, and actually you can get that green line by subtracting. So it tells us that the higher luminal glucose concentrations go, the more paracellular transport, which is not stereoselective, contributes to overall nutrient absorption. And so this graph is from one of John Medding's papers, but what Jim Madera found was that in rodents, when you did this, you got condensation of the perijunctional actomyosin ring. So this is the right mouse. So if you look here, this is a control enterocyte in a control rodent, and you see the tight junction right up here. And in this rodent that had active sodium nutrient cotransport, you see that there's condensation of the cytoskeleton around there. And I thought, well, nobody had worked on that beyond. At the time, we really didn't have tools to study this biochemically in vivo. So I developed in vitro models and found that this was associated with myosin light chain regulatory phosphorylation. And in fact, if we inhibited myosin light chain kinase, we blocked that regulation. This is thought to be important because it allows greater fluid absorption. So transcellular transport is bringing nutrients and sodium across the cell, creating an osmotic drag that brings water across, but that fluid flows through the unstirred layer above the brush border, which is really rich in small molecule nutrients because all the digestive enzymes are in the brush border. So it actually allows what's called solvent drag. The solvent, the fluid, brings nutrients with it, and that's responsible for that L-glucose absorption as concentrations get higher and higher. So this regulation probably doesn't matter. This tight junction regulation that occurs probably doesn't matter in those of us who get 3 meals a day. But if you think evolutionarily, when you might get a meal and then you didn't know when the next dinosaur was coming along, I'm kidding, I know dinosaurs and people. You might have to make sure you absorbed everything because you might eat it all at once and you need to absorb it. So evolutionarily, this was probably important. So we decided to ask, does that have any impact on physiology? So we made mice that we expressed a constitutively active version of myosin light chain kinase just in the intestinal epithelium. And if you look, this is immunofluorescence in the colon and the jejunum for phosphorylated myosin light chain. You can see it's up where the tight junction is, up at the top of the cell, and there's more of it in the transgenic mice. So it really worked. If we look at permeability using this jejunal perfusion assay, you can see the red and the yellow are the wild type in the transgenic mice. You can see the transgenic mice have increased paracellular permeability. It's modest, but they have increased paracellular permeability. You inhibit myosin light chain kinase enzymatically. It has no effect on baseline permeability in a wild type mouse, but it actually reduces that of a transgenic mouse actually a little bit below that of the wild types, suggesting that maybe there was already some compensation to bring it down, and now we've released the pressure and it went too far. So there's something going on in these mice. We have increased permeability through this physiological approach, but it's really a modest increase. If we look at the mice, this is out to 12 weeks, but we've taken it out to a year. You can't tell the difference between them in terms of weight. They grow normally, they reproduce normally, normal Mendelian genetics. They don't seem to have any defects. Until relatively recently, when a French group in several places in France, mostly Toulouse and Bordeaux, got in touch with me, and they wanted to study these mice behaviorally. And so this manuscript's under review, and what these people found with the mice is that if you track their movements, these lines are their movements in what's called an open field test. So there's a dotted line square box in the middle. That's not in the actual box. The actual box is just an illuminated open field box. And you track the mouse's movement. And what has been shown in this assay, which is fairly crude but seems to work, is that mice with anxiety-like behavior are afraid of exploring. And so they stay towards the edges, a little bit more of the box. And so when you start to look, you see the transgenic mice, actually there's a lot less lines through that middle box. It becomes really obvious. And if you look at lots of mice and you ask what fraction of total distance they spend in the center of the box, you can see that it's pretty significantly decreased. The total amount of distance they cover is not affected. So it's not because they aren't as good at walking or they are lazy or something like that. They walk the same amount of total distance. They just spend all of it around the edges. So what else might be wrong with these mice? So in anxiety-like behavior, we start thinking about anxiety and gut disorders. I think you have to start thinking about irritable bowel syndrome. And so we asked irritable bowel syndrome. We know that stress can cause these anxiety-like behaviors. We know stress increases sensitivity to colorectal distension in rodents. So we did that experiment, and we're really surprised because what we found was the transgenic mice had relative analgesia. They were relatively hyporesponsive to colorectal distension versus wild-type mice. That was relieved by naloxone. So we think this is fairly new data, and we don't have tremendous depth of mechanism, but we think that implies that naloxone is blocking descending inhibitory pathways, and so that increased permeability is triggering a response that activates descending inhibitory pathways that limit the visceromotor response. And in fact, when we look in the brain, and I'm just showing you 2 regions, but several regions lit up, CFOS, immunostain, is a marker of brain activation, neuronal activation. And if you count the number of activated neurons in the thalamus and the hypothalamus, and the same was true in the hippocampus, there are increased activated neurons in the transgenic mice. So I think there's been a lot of correlative studies between permeability and stress and visceral sensitivity and so on, and the problem with them has all been they're correlative. We don't really know what's the chicken, what's the egg. And here we know we made one perturbation to the mouse. We increased intestinal permeability by a physiologically relevant mechanism, and we got all these changes. So hopefully they will secure funding and we'll keep working on this, because I think it's really, this may mean that the leaky gut syndrome people are partly correct.
When people have studied patients with irritable bowel syndrome, do they have increased permeability?
It's such a heterogeneous disease that most studies looking at diarrhea predominant find a little bit of an increase. Find some increase in some patients. Most of the others haven't. We did an interesting study a couple of years ago where Michael Camilleri at Mayo had done this really interesting clinical trial, really, in IBS patients, where he put them on gluten-free diets and challenged them with gluten. And he found that they had symptoms that were elevated in response to gluten. And so we got those biopsies, and each patient was their own control. And we looked at things, and we saw that myosin light chain phosphorylation was actually increased by gluten. So it fits with this, but I would have to say it's all fairly soft right now. But hopefully the rest of what I'll tell you will be much more mechanistic, and hopefully this will get there and maybe allow us to do something about some of these diseases that are really hard, like irritable bowel syndrome. All right, so we decided to go for something that was a bit more of a sledgehammer, and we wanted to look at inflammatory bowel disease type diarrhea. So the first experiments we did are here. We activated T cell by injecting anti-CD3, which cross-links the T cell receptor, and it causes a TNF-mediated diarrhea. If you give anti-TNF antibodies at the same time, you block the diarrhea. And we decided to use this same perfusion assay where we put fluorescent albumin in the bloodstream, and we measure water and perfuse the length of jejunum so we can measure water transport. As you might expect, a normal mouse absorbs water, and in response to T cell activation, you end up with net fluid secretion. If you look at barrier function, how much albumin leaks through, it increases about 5-fold with T cell activation. And then we did EM, and we said, well, what's going on here? And I hope this image looks really reminiscent to you. When I showed it to Jim Madera, who had done that EM I showed you earlier of nutrient transport, he really thought it was one of his images. It really looks quite similar. And so we thought, wow, actomyosin condensation, maybe that's the answer. So we looked at myosin light chain phosphorylation, and in fact, both by Western blot and by immunofluorescence, you can see there's a lot more myosin light chain phosphorylation after T cell activation. Now I'll get into the details of myosin light chain kinase proteins and genes a little bit later, but suffice it to say, we got mice lacking this epithelial isoform of myosin light chain kinase, and we tested them. And they didn't phosphorylate myosin light chain in the intestinal epithelium. Their immune responses were normal, so it didn't interfere with T cell activation, but they didn't respond. And as you can see, they maintained barrier function, and they mostly maintained fluid absorption. Not completely, and there's another story there, but they maintained fluid absorption. So this tells us that at least for an acute cytokine-driven diarrheal disease, MLCK is absolutely essential, and this is key to barrier regulation. One key thing that was helpful was this morphologic marker. We looked at all the tight junction proteins we could think of that we could get antibodies for. This was really the most impressive one. This is occludin. And it's interesting because the occludin knockout mouse, we've recently published a phenotype of this mouse, but it's been said forever that the mouse doesn't have a phenotype, it has a phenotype, but it's a subtle phenotype, and that's in an unstressed condition. When you stress the mouse, you can see a phenotype. In any case, we saw that occludin was internalized in response to T cell activation, and this was blocked in myosin light chain kinase knockout mice. It was also blocked by enzymatic myosin light chain kinase inhibition. It turns out it's caveolar endocytosis. So in caveolin knockout mice, it also didn't occur. So we've characterized it. And if we overexpressed occludin, we could partially prevent barrier loss. So it's clear that occludin is really the player here. But for the purposes of our talk here, let's just remember that occludin internalization is a really nice marker of myosin light chain kinase-driven barrier loss. So, stressing mice. Our mice don't really have a phenotype. They have that really subtle behavioral phenotype that I don't really understand. But what about if we give them a stimulus and stress them? All right, so we use what I think is one of the better models of inflammatory bowel disease. None of the models are perfect, and we can have a conversation about what's wrong with each of them. But for our purposes, I think this is really the right model to use. Basically, we took naive T cells out of wild-type mice. We put them into immunodeficient mice. So we're putting naive T effector cells into a mouse that has no regulatory immune system. These cells go crazy and cause colitis. It's a well-established model. And we put them into either immunodeficient mice or immunodeficient mice that also had the transgene for constitutively active myosin light chain kinase. And the results were pretty striking. You can see that in terms of normal weight, they're just fine. So that's the top 2 lines. But in terms of weight loss during the course of disease, it's markedly accelerated in the mice with leaky intestines. The histology is much worse. I don't know that you have to be a pathologist to decide that this and this are really different, or this and this. So this is the transgenic in the bottom, the wild type on the top. And if you look at survival, maybe that's the easiest thing to recognize. It's a real big difference. So these mice are a lot sicker, even though at baseline they're fine. So it tells us that permeability increases can actually promote disease progression and enhance disease severity. We always try and come back and forth. I'm a surgical pathologist, so I have easy access to human tissue, and we always try to say, all right, all this stuff we're finding in cell lines and mice, does that actually happen in people? Does it matter? So these are biopsies from people with ulcerative colitis, because we could get those biopsies fixed really fast after they were taken. And what you can see is we're staining for phosphorylated myosin light chain. This bottom one's a really ugly picture, and I would never show a picture like that, except because it's terribly overexposed, except that these are matched exposures. So that's the reason for that. And what you can see is that there's progressive increases myosin light chain phosphorylation with more active disease. In fact, some patients that we had multiple biopsies with different levels of activity, at each site it correlated. All right? Doesn't mean increased myosin light chain phosphorylation causes more activity. It could be the other way around, and it could be both. But there's at least a correlation. So at least I think this tells us that studying myosin light chain phosphorylation in the context of inflammatory bowel disease may be relevant. So we took those mice lacking the epithelial myosin light chain kinase, and we did that same experiment. We tried to see if we could give them colitis. And as you might have predicted, they were markedly delayed in their development of colitis. You can see at the early time point there's a big difference in disease, both in terms of weight loss and in terms of clinical scores. A higher clinical score is a sicker mouse. But they catch up. And when we looked at intestinal permeability, we got a clue as to why they were catching up. And you can see that at early time points, they've maintained barrier function. 1.0 is a normal mouse. They've maintained barrier function. But at later time points, they both have loss of barrier function. And the reason is because we found that this immune system trigger we've given the mice is just going to go. You can't stop it. And so eventually, while it doesn't regulate tight junctions, it does damage epithelial cells and causes epithelial apoptosis. That causes barrier loss, and that allows them to catch up. And interestingly, it sort of coincides with when they suddenly drop and say, well, we really want to catch up with these guys that had barrier loss all along. So it tells us that it sort of confirms the idea that barrier loss is a contributor to progression of disease. And again, here's the histology, and again, I think you can appreciate that the knockouts, the histology is a lot better than the wild types. I think it's easy to see. So I'm going to shift gears slightly. This is a former graduate student in my lab, Sam Nall, who was really struck by some of the similarities between graft versus host disease and inflammatory bowel disease. And as I mentioned, the inflammatory bowel disease models are not great. So you have the chemical models that directly damage the epithelium. If you want to study epithelial biology, using those is just not very thoughtful. There's the immune models like we're using here. If you want to study epithelium, I think it makes sense. If you want to study immunoregulation, all by itself, that model makes no sense because there is no immunoregulation, right? So you have to choose which model you're using when. But for all of those models, we don't really know what the inciting factor is. We know they mostly don't get sick if they're germ-free. So the microbiome has something to do with it, but we don't really understand it. So we thought, wow, graft versus host disease. It's got all these similarities to inflammatory bowel disease. People have proposed some similar mechanisms, although really it hasn't been studied. And we thought, well, wow, here we know exactly when we're giving the insult. We know what the problem is. we can modify that. And so I'm just going to show you a snippet of the data, but Sam published a paper, a couple of papers. I'm just going to show you a bit of data from his second paper. And first I'll start with humans. So this is biopsies from people with graft versus host disease or healthy controls, and I think you can appreciate that myosin light chain kinase expression is upregulated and myosin light chain phosphorylation is increased. And if you look at mice, you can see the same thing. So the left column is either wild type, or in the bottom, MLCK knockout mice that did not receive a bone marrow transplant, didn't receive irradiation, nothing. They're just normal. The middle column is the mice that received an autologous bone marrow transplant. So, syngeneic, and it's basically transplant from an identical twin into an identical twin. And you can see they look pretty similar to after recovery to the mice that didn't have bone marrow transplants at all. But the third column is mice that got a minor antigen mismatch. So this is from a cousin, some overlap in antigens, but some mismatch. And the wild type mice get sick, and I'll show you that in a minute, but they also markedly increase their epithelial myosin light chain phosphorylation. You'll note that this is the epithelial isoform, and you can see that because the knockouts have plenty of phosphorylation of endothelial cells and smooth muscle in the villus core here. So it's really epithelium that's driving it, and this is the relative phosphorylation quantitatively. If we follow those mice, you can see what happens is that at day 7 after irradiation, they all lose weight, and that's from the damage of irradiation. And in Sam's first paper, what he showed was that barrier loss, if you have a mild antigen mismatch, that barrier loss is absolutely essential to initiating disease. But what I think these data show is all these mice recover, so they all had that initial barrier loss, they all lost weight from radiation, they all recovered, but then in about 3 weeks, the wild types start losing weight again. The MLCK knockouts sort of stop recovering and sort of hover. The ones who got a syngeneic bone marrow transplant, they fully recover. All right, so what we can see is that something with the perpetuation of disease is not working when you don't have epithelial myosin light chain kinase. And if you look at disease activity, the story is exactly the same. We can look at things like fur loss and stool consistency and stool blood and behavior, whether they're running around the cage or hunched in a corner. And you can see it's very clear. And if you measure serum cytokines, this is TNF, for example, there's a really distinct difference. The epithelial MLCK knockouts have less immune activation. This is the epithelial cells. So this is epithelial barrier function impacting immune regulation systemically. This is serum TNF, not mucosal TNF. And if you then look at target organs, so this is looking at small bowel in the top row, liver in the middle, and skin at the bottom, you can see that, for example, the skin, it's pretty easy to see here that you've lost the hair follicles and the sebaceous glands in graft versus host disease, and this is an area where they lose their hair. They get alopecia from it. They're preserved in the knockout recipients. You get a portal triaditis and bile duct damage, which is typical in people of graft versus host disease or of graft rejection for liver transplants. It's not happening here. And this is harder to see, but in the small bowel you get epithelial apoptosis. I think maybe you can make out those apoptotic cells, and that's really reduced in the knockouts. So I think all aspects of disease are really limited, and really what was striking to us was we had no long-term survivors in the wild-type recipients, but we had about 25% long-term survivors out to 100 days in the knockout recipients. So it really made a difference. And if you started to ask why all of this was happening, the effector cell in graft versus host disease is the cytolytic T cell, the CD8 positive cytolytic T cell. And what you can see, there's many fewer of them in the MLCK knockout mice. And then if you look, this is a marker of Th1, CD4 T cells, those are also reduced. So T helper cells and CD8 cytolytic T cells are both reduced in these mice. Any questions? No. Okay. Either you're asleep or you understand. So this is myosin light chain kinase. There's actually 3 myosin light chain kinase genes. There's one for skeletal muscle, one for cardiac muscle, and then there's this one. This one is for smooth muscle, but also for non-muscle. So we knocked it out, but we didn't affect smooth muscle. Why were we able to do that? And the reason we were able to do that is described here. So the genomic structure is at the top. Each little box represents an exon. And what you can see is that long MLCK has a promoter that's way upstream of short MLCK. Short MLCK is what's expressed in smooth muscle. The catalytic domain is encoded down here. Long myosin light chain kinase has this promoter that's way upstream. We've cloned and characterized it. It's TNF responsive, all the things you'd expect. And so what happens is long MLCK is bigger, twice the size almost, of smooth muscle MLCK. But the difference is not in the catalytic domain at all, or the known regulatory. This is the calmodulin binding regulatory domain. That's not different. Nothing seems to be different except all this stuff up here that we really don't know anything about. We know that one domain might promote binding to actin. There's been a little bit of data about some phosphorylation sites affecting Vmax of the enzyme. We don't really know when that phosphorylation happens. We really know next to nothing about those sites. But the important thing is, if you want to inhibit myosin light chain kinase, so I've just shown you data that, wow, we should treat IBD and graft versus host disease patients with myosin light chain kinase inhibitors. That would be good. The problem is, anything that inhibits the enzyme is going to inhibit both, because they have the exact same catalytic domain. So there's no way you're going to have selectivity. Why is that a problem? Well, here's a myosin light chain kinase floxed mouse where they activated Cre in smooth muscle. And so they knocked out myosin light chain kinase only in smooth muscle. And what you can see is there was massive bowel distension. The mice die of obstruction. They also have other forms of visceral paralysis. They can't empty their bladders. If you look at peristalsis, it's a flat line. And if you look at blood pressure, it's reduced. So there's vascular smooth muscle. So maybe this would be a great treatment for hypertension, except it'll kill you in other ways. So it also means we can't use that as a way to treat people with inflammatory bowel disease or graft versus host disease. That's the smooth muscle side of things. Even the epithelial side of things. Myosin light chain kinase does lots of important things, and one of the important things it does is promote cell migration. So DSS colitis, I mentioned, this is one of these chemical colitises that just causes massive epithelial injury. And then if you use a low dose of DSS, the injury is progressing, but the mice are making an effort to recover too, so it sort of attenuates the disease. What you can see is when you look at weight loss, the MLCK knockouts are doing a lot worse than the wild types. And we think that's because their migration in response to these injuries caused by the DSS is blocked. It's ineffective. So they just get ulcers that keep getting bigger instead of trying to repair them while there's ongoing damage. So it tells us that enzymatic inhibition is just never going to be a feasible therapy, even if you could just target the intestinal epithelium, right? So you could imagine oral dosing, get it into the epithelium, not let it go through. Maybe that would work. It won't work. But we did make this interesting observation. Is there a question? So we cloned myosin light chain kinase out of intestinal epithelial cells, and we kind of got lucky because the transcript's over 5 kilobases, and you can't PCR 5 kilobases all in one piece very effectively. So we did it in 5 pieces. And the piece encompassing that part of the protein, or that part of the gene, kept giving us 2 bands. And we partly might have said, it's an error, it's some sort of lab artifact, but we didn't. And we realized that what that was a very short 207 nucleotides, 69 amino acids, out of a protein that is 220 kilodaltons, so a lot bigger than that, right? About almost 2,000 amino acids. We've got an extra 69 in one version. You think, yeah, that probably doesn't matter. But we were struck by this, and we noticed that this one got turned on during differentiation in vitro. And our cultured cells that differentiate into more mature intestinal epithelial cells during culture, KCO2. I wasn't happy with that, so we developed an antibody to this region, and we did immunostaining of human duodenum. And you can see most of the staining is up in those villus tip cells. If we stain with an antibody that's down where the green circle is, that recognizes both forms, so the one that has that extra exon is called long MLCK1, the one that doesn't is called MLCK2, MLCK2 is the form most commonly expressed in all tissues that express long MLCK. But you can see that one is all throughout the epithelium. It's also in the smooth muscle. It's also in the vessels. That one's in lots of places. If we look at higher power, what you can see, not just the cell distribution is different, but the subcellular distribution is different. So if you look closely, I think you can recognize that that MLCK1 is really just up at the brush border and the apical cytoplasm right up here where that perijunctional ring of actin and myosin is. There's not a whole lot at lateral membranes. But if you look at the total MLCK stain, there's a lot at lateral membranes. So we think that primarily MLCK1 is up there in the junctional area. And in fact, when we look at colitis, and this is a mouse model, what you can see is that MLCK1 is recruited up to that region of the tight junction. That overlaps with tight junction markers in the setting of disease. And in cell culture, we could show the tumor necrosis factor drives that recruitment also. So we thought, wow, that's really interesting. This tiny little section. What we realized is that tiny little section is about half of a domain, immunoglobulin cell adhesion molecule domain, that is not complete in MLCK2, but it's completed in MLCK1. So we solved the crystal structure of that domain, IgCAM3, and we looked for, first of all, areas that were different between IgCAM3 and the 8 other IgCAMs in MLCK1. All those green circles in the diagram I showed you were all immunoglobulin cell adhesion molecule domains. And so we found this hydrophobic binding pocket, or potential binding pocket, that was unique to IgCAM3. Because we wanted to be able to do experiments in mice, we then looked for comparison between human and mouse. Now we only crystallized the human version, but we threaded the amino acid sequence of the mouse onto that and said that area is conserved. So we should be okay if we target that area with being able to do mouse and human studies. And so we used an in silico approach, a docking software, that takes the crystal structure and puts 140,000 small molecules in there, moves them around about six different conformations each, and says, how well would they bind? And, well, sorry, it's on this graph. It spits out delta G, so the free energy of binding, and those with the most negative delta G are most likely to bind. And we chose 2 groups to study, these with really low delta Gs that were rank ordered the best, and then some that were, they're still pretty good. They're in the first 1,000, but they're not great. And we thought, well, they should be really different if our approach worked, and hopefully the really good set are going to show us some that bind to that domain and perhaps block that recruitment of myosin light chain kinase 1 to the perijunctional actomyosin ring. And so we first did a screen just looking at cells with active or inhibited sodium glucose cotransport, as I showed you at the beginning, and we use those as our 2-point neuralization. Inhibition of myosin light chain kinase enzymatically brings it right up to the level of inactive sodium glucose cotransport, so we think that's a nice control. And then here's all the molecules. And you can see this green diamond here is the first one in this group. None of those changed barrier function at all. This is using transepithelial electrical resistance, which is an electrical measure of barrier function. None of these guys had any effect, but a number of these increased barrier function. So we thought those were kind of interesting. We studied them further. We had several things we wanted to make sure. First, we didn't want to inhibit MLCK enzymatically. So we did a cell-free assay with recombinant myosin light chain and epithelial myosin light chain kinase, and we added these. This thing that we know is an enzymatic inhibitor was very potent. Some of these also inhibited. We then asked, do they interfere with smooth muscle contraction? If we've targeted the domain we think we're targeting, they should not have any effect on smooth muscle contraction. Some of them did. At that point, we were left with one. We tested its ability to affect wound closure in epithelial cells, which myosin light chain kinase enzymatic inhibition would. It didn't. We also tested that if that molecule would bind to the recombinant myosin light chain IgCAM3, myosin light chain kinase IgCAM3 domain, and it did. And that molecule is right here. This is its NSC number, 55937, so there's no secrets, but that's the red diamonds. So we're calling this molecule divertin because our hope is, and I wouldn't be telling you this if it didn't turn out to be true, that it will actually divert myosin light chain kinase 1 away from the junction. And so normally, TNF activates myosin light chain kinase 1 and 2 transcription, activates them enzymatically, and activates MLCK1 recruitment to the junction where it causes occluded endocytosis and barrier loss. And our hope is that by blocking MLCK1 recruitment, we can block that phosphorylation because the enzyme no longer gets to where the substrate is. And in turn, that will block occluded internalization and block barrier loss without any other toxicities. So this molecule increased barrier function. It displaced MLCK1 from the perijunctional actomyosin ring. As I said, it didn't inhibit MLCK enzymatic activity. It didn't inhibit epithelial wound healing migration. And it didn't interfere with smooth muscle contraction. So we decided to try it in the same model. We've changed it. This was a couple of years later, so we stopped using the anti-CD3 because reviewers yelled at us about it. So now we're injecting recombinant TNF in the mice. The results are pretty much the same. So you can see is when you give a wild-type mouse TNF, you get net fluid secretion instead of net fluid absorption. If you give divertin in the absence of TNF, it has no effect. But if you give it in the presence of TNF, it blocks that. And here we're putting divertin in the luminal perfusate, so it's topologically the same as oral administration. Here's the albumin flux out of the bloodstream into the lumen. You can see you get an increase with TNF, and that's blocked by divertin.
The preventative or after the models?
In this model, it's given at the same time, so it's preventative. But I'll show you that it's also after the fact.
Are there any experiments in nature, in other words, genome sequencing or SNPs, that are associated with increased expression of MLCK1?
Nothing's been associated with expression. There are some reports of SNPs of long MLCK, not specifically MLCK1, that are associated with ARDS in African Americans. I think they're statistically pretty weak, and there's really no, as is often the case with these SNPs, there's really no biology to study because the SNP isn't an encoding region, so it's hard to understand. So there's a little bit of data out there, but it's I would call it soft. What we can see here with divertin is that by itself it doesn't really do much to MLC phosphorylation, but with TNF you get increased phosphorylation and divertin blocks that. And here what you can see is that occludin is internalized in response to TNF, and divertin blocks that as well. And then if you look at where MLCK is, first of all, I told you that the MLCK promoter is responsive to TNF. Well, the good news here is it's upregulated in TNF-treated mice with or without divertin. So divertin is not interfering with TNF responses in the mice or something like that. But what it is interfering with is movement of that newly synthesized MLCK to the junction. So here, F-actin is labeled in red, and you can see a lot of overlap. You can appreciate that here, because if we go from top to bottom and make a curve where further to the right is a more intense signal, you can see a bright peak of actin and myosin light chain kinase superimpose. But if you look at the divertin-treated mouse, you can see it's displaced subapically. And so most of it's not quite at the junction. There is a little bit of yellow, but mostly it's green and red versus yellow. So we think it is blocking that. Obviously, we'd like to know about humans. We can't do that study in humans. Maybe you guys can. Probably not. But we got fresh, normal human duodenum, and we incubated it in vitro with TNF, with or without divertin. And we got really the same results. The images aren't as pretty because this tissue is not happy. It's devascularized. But you can see that MLCK1 expression is increased. You can see that it's displaced downward from the perijunctional actomyosin ring if divertin is present. And there it is quantitatively. You can see that myosin light chain phosphorylation occurs and is blocked. You can see that occluded internalization occurs and is blocked. So it seems to do all the same things in human tissue as well.
Have you tried it in something like an organoid model? Something like that where you have like a 3D cell culture?
What would the question
I'm just
be?
wondering, just because based upon like similar with the ex vivo, that if you're going to have like kind of that luminal, and then you're still going to be able to see.
But the other was the mouse was in vivo. So the problem with organoids, yeah. So I'll give you my opinion. The problem with organoids is that everybody says, oh yeah, they're differentiated, and when you look carefully, what they're showing you is mRNA that tell you they have some differentiated protein expression. They're not differentiated. Only one group that I know of has differentiated them so that they transport. That's the Donowitz group at Johns Hopkins. Mouse organoids, nobody has differentiated so that they function and transport. And so really, although it's unpopular and it's definitely not on the bandwagon, I think Keiko2s are a much better model of absorptive enterocytes than any enteroids. I'm trying hard to make mouse intestinal epithelial cells grow as monolayers that we can do transport in, because we have all these interesting mice, and at present we do is we make the same mutation or knockout or transgenic in mice and cell lines. And it's twice the work, and it's still not the same thing. And people have this bias against cancer cell lines. But the fact is there's a few cancer cell lines, like Keiko2, like TD4, out of the thousands of colon cancer cell lines that exist, that people use. And there's a reason people use those. It's because they behave really like normal epithelium and can be induced, if you handle them correctly, to behave like real transporting epithelium. And so we've tried, we can get monolayers to reduce their barrier function in response to TNF, monolayers of enteroid or collonoid cells, but it's because the cells are dying in response to TNF. We cannot get them to regulate their junctions. So does that mean the Keiko2 result where we see them regulate their junctions is an artifact? No, we see it in vivo, we see it in mouse, we see it in human. It's definitely correct. It's definitely what's happening. We just can't get the enteroids to behave quite right. And if you think about it, the way we grow them, we're stressing them. When we grow them, we're growing them in high growth factor media all the time. You take them out of growth factors, they grow much less well, and then they apoptose, which you'd expect them to if they mature and migrate up, right? So the conditions we grow them in are not really very amenable to differentiation. And people have tried different protocols, but I don't think anybody's really got it right yet.
But you've used mouse organoids for other purposes.
Oh, for sure, for sure. Yeah, we use them in my lab.
Yeah.
We definitely use them in my lab, and they're useful for what they are. But I think if you're looking for a differentiated function like this, they're probably not the right model. Just like DSS colitis, I don't think this would help very much in DSS because there's no tight junctions to preserve. There's just damaged epithelial cells, right? So back to those healthy relatives with increased permeability. If we gave them Divertin, could we collapse them down and prevent them from ever developing disease? Or even better, if we took these patients with increased permeability and treated them with Divertin, could we keep them in remission? So that was the first experiment we did. We gave mice a T-cell transfer, and then when they started, just before they started to get sick, but we knew they were going to get sick, started giving them Divertin. And it was really effective. When we sent it off to the journal, the editor said, yeah, but people don't come in saying I'm going to be sick next week, treat me now. I think in the case of IBD they do, because we're talking about maintaining remission, so it's really a different game. But the editor's always right. So we did that experiment. So here what you can see is we had 4 groups of mice, and we had mice receiving, actually 5 groups. So the white circles are mice that did not receive T-cell transfer. And then the 4 colored groups all received T-cell transfer, and you can see right there at about day 20, the weights diverged. And when that happened, we started treating. So we waited until they showed clinical evidence of disease, and weight loss is, as you would expect, is a lagging indicator of disease. They've already got real disease if they're losing weight. We start when they're all pretty young, and so that's why you have to have the controls because they're still growing. We start when they're 6 weeks old, so they're sort of adolescent mice. So you won't know that they started losing weight if you just see it sort of flatten. So the purple is Divertin and anti-TNF together. The red is Divertin. The blue is anti-TNF. So anti-TNF, Divertin, and anti-TNF all worked pretty well for a little while. They all restored barrier function acutely, and the very next they all helped weight gain pretty quickly, and they all worked pretty well for a while. But eventually, anti-TNF alone didn't do too well, and we know that's true in this model, and we know that's true for lots of patients. The clinical scores are exactly as you'd expect, and survival is exactly as you'd expect. So Divertin and anti-TNF together were better than anti-TNF alone or no therapy. If we looked at colonic length, which in this model isn't a great indicator, but if you look, you can see there is some colonic shortening with disease, and that's blocked by both Divertin and Divertin plus anti-TNF, but it's not blocked by anti-TNF alone. If you look at intestinal permeability, all of the therapies restored barrier function to some extent. If we looked at stool water, so in this graph we have an extra group. We have the orange group of mice. Those are mice the day before we started therapy. So you can see they've got increased stool water. So you could read that as diarrhea right before we start therapy. In the mice that received saline, that continued to increase. In the mice that received Divertin, it decreased. In the mice that received anti-TNF, it didn't increase as much as in the mice that received saline, but it increased more than the mice that received Divertin. Same was true, mix or match for the different cytokines that we looked at. Some were affected similarly by Divertin and anti-TNF, some were not. So for example, TNF and interferon gamma were affected by Divertin, but not anti-TNF. IL-1 beta and KC, which is IL-8 for a mouse, were affected by both. So biochemically, there was some effectiveness to both, but in terms of weight and everything else, you'd say, wow, Divertin was better. And if you look at the histology, I think it becomes really clear. So histologically, there was really very little difference between saline and anti-TNF by the end of the experiment, versus Divertin still preserved. You can see there's preservation of goblet cells. So those of you who looked at IBD histology with people have heard of mucin depletion. It's not so much here. We don't have crypt abscesses, which we have plenty of in the other mice. This is one of those mice the day before we started therapy, so it looks pretty sick too. So it really helps. So I hope I've explained to you how intestinal permeability amplify nutrient and water absorption. So they're part of our normal physiology. So increased permeability isn't all bad. And if we had another hour, I could tell you about how increased permeability in the appropriate context helps you clear pathogens. So it's not a bad thing always. And intestinal barrier defects are insufficient to cause overt disease. And until recently, I would have ended this statement right there. But I've had to add now that it might induce more subtle visceromotor and stress responses. I can tell you, I didn't show you the data, but if you look at those mice, if you breed them a transgenic male and a wild-type female, so half the pups are transgenic, but all the moms are wild-type, and then you look at the microbiome, they also segregate. So there's some effect on the microbiome too. Intestinal permeability, I hope you understand, can be a target and an effector of mucosal immune regulation. TNF can cause barrier regulation, but barrier regulation clearly can help drive the immune response. And myosin light chain kinase is that key signaling node. So we can target it and prevent acute and chronic disease-associated barrier loss. We can prevent progression of experimental disease. MLCK is activated in lots of other intestinal barrier defect diseases like infection. So there's a lot of potential utility if we were able to develop such a molecule. And I think this idea of blockading recruitment as a non-enzymatic approach is effective, and it both prevents and reverses experimental IBD, and it has incredible promise, because I think the idea of not suppressing the immune system, which at the moment all of our IBD therapies do, and all of our GVHD therapies do, the idea of not suppressing the immune system, but restoring the barrier, if not replacing immune suppression, which I don't think it probably will, it can certainly synergize and perhaps reduce the doses of those immunosuppressants required. And you can play a semantic game and call them biologics, but they're still immunosuppressants, and they still do bad things to people. So from the MLCK knockout mice, you would guess that this would be pretty benign, this treatment. You can measure permeability really easily. You just have them drink a little bit nasty tasting solution and collect their urine, and you see how much showed up of lactulose and mannitol. So it's really easy to do, can be done by mail order. So you could monitor patients and say, wow, I think we need to, we're seeing a little bit of tick up in your barrier, in your permeability, maybe it's time to intervene and prevent what we know fouls barrier defects. And then hopefully, I don't know of any approaches where people have said, let's prevent an enzyme from getting to where the action is. We're not inhibiting its function. It can still do all its other things, but it can't get to where the action is for this one thing we don't want it to do. So hopefully that might be a paradigm for a new approach to therapeutic targeting, because really biology is and evolution are pretty clever, and they use the same enzymes for lots of things. So just blocking them entirely might be too crude in many situations such as this. So I like this picture. I took this in Botswana, and I pretend that these guys are all working hard, and this guy's just talking about stuff, and this is me, and this is the lab. Mostly I fix things in the lab, I help with microscopes, but these are the people who do the actual work. A few of them are here. These are people who did some of the other work that I've shown you, who were alumni of the lab. Collaborators are key. Always try and find good collaborators. You can't possibly know everything. And obviously, without financial support, we close the lab. Thanks.
I think I mentioned I overlap with Jim Madera quite a bit at Emory, and he's actually the president of the American Medical
Right.
Association.
And he likes it. I can't imagine. But he really likes it. And you know what? We as a country need somebody like that as CEO of the Emory right now.
Yeah.
Somebody's really promoting. He's doing great things there. I told him it was almost enough to make me want to join.
So the graft versus host disease, is it likewise, again, you probably mentioned it, related to TNF-alpha expression and its effects on tight junctions, on these barrier junctions?
Well, I think
or
that's
MLCK?
what we're showing in the MLCK knockouts, right? What we did, I didn't show you, so you can rightfully criticize us. That is a whole animal long MLCK knockout. We can't exclude that long MLCK expressed in other non-muscle cells is responsible for the phenotype. So what we did, we've tried to make an intestine-specific MLCK knockout, and it doesn't seem to work. So instead what we did was we put our constitutively active myosin light chain kinase, which is only in the intestinal epithelium, onto the knockouts and asked, could we complement back wild-type behavior? And we could. So I didn't show that figure because it's more complicated to explain, but it's in the paper. So we are pretty confident that it's epithelial myosin light chain kinase that activity is the key, and we think it's tight junction regulation.
Yeah, great. It's fantastic work. So is Therian, is that the name of the startup?
Ethelium, like endothelium, epithelium.
Is it working on Divertin-related molecules?
Yeah, so it's a tiny little company. There's one person who holds all the titles, Valen Graham, who was the first author of the Divertin work. He started it as a graduate student years ago. That was a project that took a long time to mature. He's done a postdoc, decided he doesn't want to be academic, wants to be an entrepreneur, and in putting the paper together, he said, I want to start a company about this, and I was happy to because I hope you can appreciate the assays are really pretty subtle, nuanced, and complicated, and we spent 25 years figuring them all out. I don't think very many other people would be equipped to pursue this because they don't have that experience. And so Valen wanted to do it, and I thought that would be a great idea. So we've started this little company. He won a gold ticket award at one of the things in New York. He's in New York, so he got free bioincubator space for a year. We got an SBIR from a small business grant from the NIH, which is for a small amount of money. And the approach we've taken is to use our crystal structure, use Divertin. We have two other small molecules that we haven't taken as far as Divertin, but as far as we've taken them, they do the same thing. And so using those Divertin and those two molecules, we've modeled the interaction at a much higher resolution. And now we've run a screen and come up with 100 molecules that should bind pretty well. And so the grant is to do the in vitro studies of those to ask if they increase barrier function, if they TNF-induced MLC phosphorylation in KCO2 cells, which I didn't show you, to show if they block MLCK recruitment to the junction, to show if they don't inhibit myosin light chain kinase in smooth muscle or the enzymatic activity. So that's phase one. It's a small amount of money. It's for a short time. And that's where the company is. So unfortunately, Divertin itself is not suitable for the standard medicinal chemistry stuff you'd think of doing, where you like add a group here and change a molecule there. It's just too small. So our goal is to identify things that are more drug-like.
Yeah, that's fantastic. And just one last question. The sort of Anno's lactulose assay to assess intestinal permeability, is it used clinically at all?
No, but I mean, there is a CLIA certified lab in, I think it's called Rocky Mountain Labs or something. If you Google it, you can find them. And I think for about $100, they'll send you, and you can send them your urine, and they'll assay you. Yeah. And maybe there are some physicians out there in private offices sending off, but as far as I'm concerned, there's no justifiable use for the assay clinically right now. And actually, I think we can probably do better than that assay. So lactulose and mannitol are really close in size. So this ends up being a sort of wonky discussion that I'll stop. But I think we can probably do better, have better probes than those. That's just what's been used historically.
So what do you need to take this to clinical trials in humans?
We need a molecule that could be a drug. So in our experiments in the mice, this is a really small molecule. I could draw it for you, but it's a really small molecule. We don't have any idea, but everybody who knows something tells me it's unstable in water. So what we did in the mice was we dosed them. We found that 25 milligrams per kilogram was the max tolerable dose. That's what the NCI found also. The library we screened was an NCI library where they had years ago and given them tumors and screened a bunch of agents, a bunch of small molecules for anti-tumor effect. And with this one, what they found was that at 25 milligrams per kilogram, every day, all the mice survived for 30 days, but none of them had tumor responses. So they threw it out, but it's in their library, and that's the library we screened at the time. So what we need is something, and we don't have any way to follow it, so we just used the max tolerable dose and crossed our fingers. So we really need to have a molecule that can be given to humans, that we can follow, that has higher affinity. In vitro, the doses of this required are about 150 micromolar. That's at least 2 orders of magnitude, at least 4 orders of magnitude more than. We gave both, in the chronic disease model we gave IP, but in the acute TNF model we gave intraluminally. So it'll work either way. But 150 micromolar, you're usually talking about things in the nanomolar range for real drugs.
I know, but they have so many really interesting nanoparticles that you can encapsulate and then have it line the entire intestinal tract. All
But you'd have
your friends over in surgery that are doing that
Yeah.
at Brigham.
You'd have to let it get released there and have it cross the cells, and we know it will from the perfusion studies.
Really
But we don't know how
doses.
stable it would be.
Why don't you talk to Ali about that, Ali Tavicoli, and see and encapsulate that into his nanoparticles and then coat the whole bottle with it and see if it works.
So we know that it works in mice the way we're doing it, right? To do that, you'd still be in mice, and at the end of the day, you'd still have an unstable molecule that you don't have tox for. So you still couldn't go in humans.
I guess you also have the problem of diarrhea washing away.
Yeah, absolutely.
If the patients come in with IBD and diarrhea, then it's washing away.
But we know the mice had increased water content in their stools when we started dosing them.
Yeah.
But that was given systemically. So I think really what the advice I've been given, and I'm a novice at drug development, and that's why we're doing it in a company. My lab is not doing any drug development. We're trying to understand the biology of this more. And we may come up with new drug targets and understand the biology, but we're not trying to find small molecule drugs. I don't think that's what we do in an academic lab, at least with what I know.
Yeah, but this is a great example of fundamental molecular biology, great structural biology, medicinal chemistry, and also this sort of new paradigm of sort of altering sort of intracellular shuttling and transport, of complexes, to impact sort of the end function of the assembly, molecular assembly, right?
That's what I hope. That's what I hope. I mean, we're really excited about it. They granted really, really well. Valen is the name of the guy who I'm allowed, because of Harvard rules, I can't be anything in the company other than a shareholder and a co-founder. So he's the CEO, CFO, CSO, anything you want, and lead technician. But Valen has presented it to all sorts of venture cap things in New York, and he always gets a really strong reception. So I think, there's incredible promise here, but we do have to take it to something that could be a drug.
Any other questions? Fatima.
Hi.
Question? Fatima is one of our wonderful alumni who's actually finishing up her pediatric surgery fellowship at Children's Hospital and joining their faculty.
Oh, great.
And a great
I was
investment.
hoping to talk to you offline.
Absolutely.
Questions and thoughts.
Absolutely. Do you know Kathy Hunter and
I do. I've read some of
and the whole CHLA group?
Yeah, and some of the work you've done in NEC is really interesting.
Well, no, I didn't do any. It's
Well,
all
you helped.
Kathy was my mentee. It's
Yeah.
all Kathy.
Kathy did it, but anyway, I'm hoping to chat.
Yeah,
Great.
that would be great. That'd be really great.
Dan, any questions?
I understand what you're saying about, is there evidence that TNF is the sort of culprit in non-immunopathology driven insults to the colon? So for instance, ischemia, surgical anastomosis, things where you would want to enhance tight cellular, tight junction, but it's not primarily driven by
So ischemia, I think, is driven by cell death. I mean, there's this characteristic histology that I see as a pathologist where the superficial cells are damaged and the crypt cells, if the ischemia is not too bad, are actually hyperproliferative trying to repair. So that's not going to be a tight junction thing. It could be affected by tight junctions at some level. I was very resistant with the editor. I tried really hard to talk her out of making us do that last experiment because A, we know TNF isn't great in those models, so I knew Divertin, if it worked, would do better. But I was really afraid that Divertin wouldn't work once there was damage, and I was wrong. So we got lucky. I think ischemia, there's arguments, and we could have a whole nother conversation about occludin. We found that occludin, in and of itself, downregulation of occludin prevents apoptosis, which is really surprising to us. So there's other avenues, but I don't think TNF is the primary effector in the two examples you gave. In wound healing, like anastomoses, that's not tight junctions. Eventually they have to be assembled, but first it's cell migration and that sort of thing.
Well, thank you. This is wonderful. I have some gifts for you.
Oh, thank you.